toxicity, biohazard, but as well in the heterogeneity or in the purification of how pure is your sample. So basically, you as a user, you most of the time you know what it is. So you have a lot of a priori knowledge. Is it a liposome, an EV? Is it a virus, a vesicle, or technical particles such as quantum nanodots or spion particles or artificial particles? But the question is, do we have aggregates? So what's the quality? Do we have participates, nanobubbles, things which are not in there, but we have to consider what it is in order to say something about purity. And today I guide you a little bit with NTA, what we can do with NTA to cover exactly those aspects of homogeneity and purity. So nanoparticle tracking analysis is a quite straightforward technique. You inject, you visualize. It's a microscope. It's an ultra microscope, so the particles appear in white and you have a black background. And then, based on the analysis of such videos, we measure, we gain data. We gain data from that based on this setup. So the laser is perpendicular to the objective, so that's the laser, and the objective magnifies the nanoparticles. And basically what you see on the screen, it's a 2D reflectance pattern. And 100 years ago, an Hungarian, Austrian physicist named Zsigmondy, he came up with this brilliant idea to do this perpendicular. And this is called an ultramicroscope, and this is known of today. So this is actually nothing new, but 100 years have passed, and Mr. Zsigmondy got his Nobel Prize. And with the help of nice lasers and nice sensitive cameras, we can really get nice data out of such a microscope. And basically, particle metrics started out with measuring zeta potential, and we found out very soon that we also can do size and count because we know the volume. And just lately, we also applied, since 3 years now, fluorescence in order to gain specific data from our sample. So this is how NTA technology works. Basically, it's a video. We take a video, and detection and localization of the particles is done by means of an algorithm, by automatic processing of the videos, and then we quantify the average mean square displacement of the particles, basically the jump the particles do from frame to frame. And if we link this together, and if we apply Stein's-Einstein equation, so we get the diffusion coefficient, and out of the diffusion coefficient we get the radius. And it's a single particle technique, so each single particle is, so the size is determined, and of course, if you want to have a size distribution, you do this based on statistics over a lot of particles then. And those are the parameters the nanoparticle tracking can do. It can do the size, the concentration, the zeta potential, and the fluorescence. So how can help fluorescence NTA, or how does it work? Basically, when you measure in scatter mode, you see all of the particles. So the laser comes, it creates scatter light, and the scatter light is directed to the camera. When you have the fluorescence mode, basically, we put a filter here in order to block the light of the laser, and only the fluorescent light is transmitted, and the laser light is blocked. So we measure the fluorescent particles, and so the concentration is of the fluorescent particles. And this gives you now the aim with the help of fluorescent dyes. You can do vesicle ratios, biomarker ratios, so you gain specific data. So this is an application from liposomes. They are stained with the dye Dio, and we use the 488 nanometer laser to excite the fluorescence. And as it is a fluorescent commercial product, so we gain up to more than 80% of the liposomes which are bearing such a fluorescence. So here you can have some staining of the membrane, and it helps you in order to detect those liposomes in the presence of perhaps nanobubbles or stuff which is not a liposome, which doesn't bear a membrane. Basically, in general, you see an overestimation because you also measure your air bubbles, your participates, or whatever it's in there, but which is not your analyte you're actually looking for. And by vesicle ratios, basically vesicle concentration versus scatter concentration, it gives you a ratio which you can use as for quality assurance. So those are the principles we can apply: fluorescence, basically we have the scatter mode. We measure, we call it the so-called total particle count. Then by use of membrane dyes, which we just saw for the vesicles, we get membranous particles, and so there are a bunch of dyes out there which works. And then by use of an antibody, we can even gain more specific information as the antibody binds to the surface, to the epitopes, and the antibody bears also fluorescent fluorophore, and the light from the fluorophore can be detected. And even we can do secondary antibodies, of course, which gives us much more freedom for our analysis. It comes to the critical question of the labeling protocol, and here we can derive a lot of information from flow cytometry already, because they have all the antibodies, they have all the fluorescent dyes, and basically we have to adapt it to NTA a little bit. We have to talk about the quality of the antibodies, and so there will be new products in the markets dedicated to NTA technology. Basically, you have the antibody and the vesicles, your bio-nanoparticles, you incubate, and then you dilute to make it ready for NTA measurement. And the question is if you have to wash or not to wash off the free unbound dye. And this depends on your application, of course, but both ways work. So let's shift gears a little bit and talk about our main applications, which are the EVs. EVs are shedded particles from cells, basically very small particles in the range from roughly 30 to 100, 150 nanometers. And of course, with the instrumentation, we also see the microvesicles, the EVs. And by means of antibodies, we can distinguish what it really is. So this was done with our project partner in Salzburg. Basically, those were MSC-derived EVs, and with the help of different directly labeled antibodies in sequence. First, we measured the CD63 concentration and the EVs bearing the CD9. And so we get kind of an, I would say carefully, as kind of a phenotyping of the surface. And the same was done with another project partner. Here we did EVs from transfected HAK293 cell type. So basically they're using a special antibody, what they were using in order to detect if the transfection of the cells was correct and if the EVs also bear some of the information of the transfected cells. And you here see the wild type, so the CD40 and the GP350, they are typically not on the wild type, whereas if you do the transfected, the analysis of the transfected cells, you can see that those are really bound, so the antibody bounds on the surface, and so you can see if the transfection was working. And of course, again, we add specificity for NTA technology. So this gives you now additional parameters which you can use for characterization and quantification, getting a quality of your product, such as a biomarker concentration, biomarker to vesicle concentration if you use a membrane dye, or if you have a ratio of different biomarkers, which can be, of course, for you, it's not the CD63, but different types or different biomarkers what you use perhaps for special diseases. So this can help you in the development of new pathways for diagnostic, for example. And if you have dedicated antibodies, it's also possible to specifically detect virus within, your reactor. So you can detect specifically virus by means, here it was an example of a secondary antibody, but if you have a primary antibody, this also works. So specific virus detection. So this was nice, what we saw before. So this was done with a single laser. Basically, we have to measure one biomarker after the other. So next generation, then the next step is we just combine several wavelengths so we can measure on the same sample several biomarkers. So this is now our new twin system. Basically, we measure the scatter, we use laser 1, and then we can shift with the wink of an eye with no alignment between the lasers. So this gives us now the option in order to measure 2 biomarkers on the same sample. Again, the power is in the antibody. You need to find a nice antibody with a strong binding constant in order to see really biological information. So we did an antibody evaluation, so we used different clones and different dyes, and it's really the power in the antibody. Of course, this has to be assessed, but this is not new. We also know this procedure from flow cytometry. So basically, if you have single wavelength, you have 2 samples, and you measure CD9 and CD81. So you need 2 samples, you need actually 4 measurements in order to do 2 biomarkers. And if we switch now with our twin system, so we have the CD81 on the blue channel, and on the green channel we have the CD9. So basically by switching, we can measure the 2 biomarkers on the same sample, and this is actually data we present for the first time, basically in sequence 2 biomarkers within 3 minutes, which is quite fast. So the data is in good correlation. Basically, if we compare those 2, basically with single wavelength, you need more time and more sample, and with 2 wavelengths, you're faster. So, and this was done also for HAK 293 derived EVs, and we also could show that on 1 sample, we can do a double estimation right away with our system. So there are, we have several wavelengths available, and there are several dyes, and basically stable dye is recommended for the use of directly labeled dyes. And the nice thing is you can also use PE, which is quite commonly available, or APC, which are actually already commercially available. So you can use commercial available antibodies in order to have specific information. Yeah, this is a nice image from one of our demonstrations in Asia. So our instrument is rather fast, so we measure a lot of samples in a short time, which is crucial if you are working in a laboratory close to a clinic, and yeah, so time is key. So concentration, we know because we know the volume. It's a single particle technique, so we need to gain data in a short amount of time, which we can do. So don't believe only on 1 position measurements. So we need statistically sound data, not to think we have extremely high resolution, but we really need to see the whole sample in order to get a representative picture of our sample. Also, talking a little bit about concentration assessment. We use a concentration calibration material, and this is key in this case. We calibrate our instrument, and you as a user decide what you use for calibration. And basically, there are different calibration materials out there, and we checked 4 different vendors for so-called concentration standards, and we were very amazed. So there were variations up to 60%, and this is also a question to the vendors for those calibration material, and so we have to talk. Why is this happening, and how can we make products better? So we need both the standard and the instrumentation. High resolution scatter plots, additional data. So that's the NTA, and we have CEDA potential measurement, or for hazardous samples, so we have different cell assemblies available. So fast injection, fast cleaning, so it's a fast instrumentation. Basically, those were the analytes on the application. So for virus concentration, which is specific, we have the virus titer, grade of purification, phenotyping, concentration, with the extracellular vesicles, which are very promising in the medical market, and with the liposomes concentration up to stability if you include CEDA potential measurements. So those are very typical applications, and I'm more than eager to hear about your applications. And even more, so besides the bio-nanoparticles, we go to also the technical products, of course, nanometals, nanobubbles, quantum nanodots, spions, and much more particles. So those are the 4 parameters for NTA: size, concentration, CEDA potential, fluorescence. And let me sum it up. It's a fast technique, 2 wavelength in sequence. There's no alignment needed. So this is a quick switch between the wavelength and the laser and the fluorescent mode. So fluorescence adds specificity to your sample. It's very good, very low variance inter and intraday, and it's very sensitive due to a low bleach technique. So even we see GFP as a nanolight. So we would like to thank our cooperation partners from University in Essen, from Lonza, and from Hansa Biomed in Salzburg. And part of the work you have seen here was funded. Thanks a lot for money and all the cooperation partners, and you for listening to my talk.
Thank you very much, Clemens. The floor is open for one quick question, please.
For the twin laser system,
yes.
Can you measure the intensity of a particle with both wavelengths at the same
In sequence, we can measure that in sequence, and we also can measure the intensity of the particles so we can compare.
So, but you look at a distribution of both.
Yeah, yeah, we look at the distribution of both.
Can you get it to the level where for a particular particle that you see both at the same time?
So you're talking about co-localization, so on the same particle with two lasers. We definitely work on that.
Thank you.
I have many questions, but I'll leave them for later when we have seen all the instruments and all the presentation. So for the interest of time, I will, thanks again